share with you the fact that this is a rare opportunity that some of you from all over the country have been selected to participate in this program, which is going to be inaugurated today by Dr. Shekhar Mande, Director General of CSIR and Secretary DSIR, one of the most renowned scholars, and I will introduce him at 10 o'clock formally. Uh, you should also be proud that the valedictory function of this school will be on 21st of December under the chairpersonship of Dr. Syed Hasnain, Vice Chancellor of Jamia, and the valedictory address will be given by Dr. Bhargav, who is the Director General of Indian Council of Medical Research, leading the entire struggle, entire war on corona that is taking place in our country, Secretary of Department of Health Research, and therefore two of the luminaries of our country would be addressing you on the first and the last day, but then during the program also you will be meeting, interacting with many scholars, scientists, and other specialists. Why are we doing this? What is the motivation for us to do that? Why Honeybee Network, SRISTI, GYAN, and all other institutions together are participating in this? Why has SRISTI, which also has a natural product lab, as Megha might have mentioned to you, and where we hope some of you will come and work in due course when corona adverse effects are overcome. Why we are doing it because we believe that young scholars of our country, young scientists of our country, the budding leaders of our country, of biotechnology, of life sciences, should take interest in the knowledge system of our society. Should recognize that this country, this society has survived for thousands of years because of some innate strength in our ability to identify the problems and solve them. So as you might know, that Honeybee Network started 33 years ago, began by giving voice, visibility, and velocity, voice, visibility, and velocity to the ideas of creative communities and traditional knowledge holders from all over the country. I'll describe to you briefly about various activities that we do, but it is important to mention that we expect during this BIIS, each one of you to review literature on traditional knowledge as well as contemporary innovations developed by farmers, pastoralists, horticulturists, and other food processing entrepreneurs, or fisheries for that matter, depending upon the background that you represent and the interest that you have. You will develop research protocols, assuming that you are going to work on that experiment tomorrow to validate and value add in a people's knowledge, then you will develop a protocol. And we want that best of the science should serve our society. We should not say that if it's the knowledge of common people, then we can do a second grade job. No, we will use the best possible science to identify the challenges that we have. To illustrate, and this experiment I did recently in my own home, so I can tell you with confidence that it works. But of course, we have tested it many times before. We published in 1992, an article by Dr. Chari, former director, Central Tobacco Research Institute in Andhra Pradesh. And he reviewed a literature on the use of milk for viral control. So what farmers used to do, when they will transplant seedlings of tobacco, they would, tobacco is not good for our society and for our health, all that is true. But the fact that this crop was grown at one time and it still is grown by a large number of farmers, we need to understand the significance of this practice. So when they transplant seedlings, there is a virus that affects tobacco. Which is that virus? Anybody knows?
Tobacco mosaic virus.
Huh?
Tobacco mosaic
Sir,
virus.
it's TMV.
Okay, and which virus is it? DNA or RNA?
Virus always is an RNA, sir.
Huh?
Virus always is a RNA, sir.
Which RNA? Type 1, type 2?
Virus cannot be always a RNA for you, Laksh. should learn.
You should clarify that to him. You should clarify that to him, that it can be both. But this one is type 2 RNA virus. So I'm happy, and this culture, this spirit of arguing with each other should continue in the entire course. Nega, you should encourage students to argue with each other, contradict each other, fight as strongly as they can. If there's no fight on a particular day, then we have not had any energy in the course. So there should be a lot of fights in the class.
Definitely.
Intellectual fights, of course, we don't have to fight otherwise, but intellectual fights, strongly, assertively. And you should then provide evidence for your argument. So who spoke about the virus not necessarily being RNA virus?
Me only, sir. I was saying that mega.
So why don't you? So my suggestion is that we should put down on the chat simultaneously some references, so that whenever we make a point, and the person who said it is RNA virus should also put down the reference on the chat, so that a dialogue starts right away. The very first day, the very first session, we should begin to talk to each other through evidence, through evidence of prior literature, so that the discussion becomes more evidence-based. Our curiosity should not become fueled by our passion, which is hollow. It should be full of our passion, which is backed up by good science. We will not compromise on science. So coming back to the practice. So what do people do? They used to put a pot of milk alongside in the field when they transplant seedling. And after every few seedling, they will put their hand in the milk pot. So assumption was that there is something in the milk which inactivates the virus. So the new seedling that they will transplant would not be affected by the virus on their fingers, on their hand. In Gujarat, we have a practice, and there's a database you can look at. Please write down meghasashti.org/WSA. And there are 5 databases there. You can access them freely, openly, and you can select some practices even out of this database, not necessarily by only among the one that we give you. If you want if you find some practice which is intriguing to you, you can as well take that up. So what is done now? So then in the North Gujarat, when they have foot and mouth disease, is it a bacterial disease, fungal disease, or viral disease? बच्चों, foot and mouth disease किसी को पता है?
So I think it's prions.
चेक करो बच्चों, थिंक नहीं, अभी चेक कर लो ना अपने पास. अब नहीं मालूम है तो चेक कर लो. FMD कौन सी डिजीज है देखो. ये ओपन बुक एग्जाम में कोई दिक्कत नहीं है, पूछ सकते हो, चेक कर सकते हो, पढ़ सकते हो. बताइए क्या है ये?
वायरल डिसीज.
वायरल डिसीज.
और कौन सी वायरस है ये? बताओ.
तो
Picorna virus.
It is also type 2 RNA virus, ठीक है ना?
Yes, sir.
So, and when I was a child, I had a pain in my ears in the 1950s, late 50s. So the landlady put her breast milk in my ear. So I shared all these 3 observations in a conference on biochemistry and plant physiology in late 80s. Dr. S.K. Sinha, at that time, professor of plant physiology at IRI, was the chair of the conference in Kottayam. He had invited me because the conference focus was on drought, and I had done a lot of work on drought, coping with drought by the farmers. So he said, we know about how plants cope with the drought. You tell us about how people cope with the drought. So I had gone there and I shared that. And after that, I mentioned these 3 observations. And I said, what is the connection? Can any one of you see the connection of these 3 observations in context of what I've said? Using milk to inactivate, supposedly, assume that it actually inactivates, but that's what farmers practice. So we as a scholar, we would not believe this practice just like that. We will have to subject it to the experiment. We have to subject it to the review literature. We'll have to develop a protocol to validate it. So what's the connection in these 3 things? क्या कनेक्शन हो सकता है तीनों में, 3 ऑब्जर्वेशन?
Sir, mostly, probably I just can assume that we can say that from this all the three observations that maybe milk has some constituents related to the virus which are leading to the slightliest inactivation of the viral.
So what are the mode of action? What could be the mode of action? Let us assume for a minute, let us speculate what are the pathways through which a virus can be inactivated broadly. So virus is in which envelope?
So protein envelope.
Huh?
Protein envelope.
Protein envelope. So what do you need to cut into protein?
Protein,
Protease?
yes, sir.
All right. So does milk have protease? Check karo.
Yes, sir. It has lactase.
So it can break into protein. प्रोटीन वॉल को ब्रेक कर सकता है कि नहीं कर सकता? हिंदी में बात कर सकते हो अगर किसी को हिंदी बोला है, तमिल में बोला है, तमिल में नहीं समझता हूँ, but that's a problem. How many of you don't understand Hindi at all? Anybody in the class who doesn't understand Hindi at all? So I should not slip into Hindi. Are there students who don't understand Hindi at all? Anyone?
Yes, I am.
Okay. What's the name?
Sir, Nitish Kumar Hall.
Nitish. Okay, I'll take care. So now tell me, what could be the next step after you cut the protease cuts the protein code? What should be the next step? Which enzyme could snipe the virus, RNA virus?
RNAs.
RNase enzyme. Now look at this. You may be undergraduate student, but by applying your mind, you are attacking a problem at the heart of the science, where for such a long time, scientists have actually not paid enough attention to my mind. They could have. And I will share with you a blog there where we have put a lot of references on use of milk for viral disease right from 1935 onwards. And the point I'm trying to drive home is that this is a good science. Just because people did not know the reason doesn't invalidate their knowledge. Sometimes they may do right thing for wrong reason. I will give you an example of that also. But the question is, unless we have curiosity to subject our observation to a critical scrutiny of science, we could get carried away by our belief system. And I don't like that. So Saksham, you should write now, Saksham and others who have commented on this, you should write a short note with at least 30, 40 references tonight. Three of you or four of you, you can choose. Mega, please put them into a group. And those who have spoken on this subject, your responsibility is to educate the entire class about the processes through which milk could help in activating a virus in animals, in plants, and perhaps in the human beings. Now, which viruses can be inactivated? What will be the process? Please put down your names. Say those who have spoken on this subject, they can volunteer. And tonight you will write a review, and tomorrow, Mega, give them some time to present it before the class, okay? Make adjustment in the calendar. After all, every day we should get one review made by the student team, those who take initiative and would like to, who have an observation, who have a keen eye to observe some relationship. We should encourage them, not the same group, different groups, different days, all right? So everybody should get a chance. Every one of you will get a chance during these 3 weeks to present, among other things, a review on a topic that comes up in the discussion in that day, on the day, next day in the class, so that each one of you work hard. There will be many more things that you have to do, of course, but I'm just mentioning one exercise. So what are we trying to say? We are trying to say that when you will work on, let me show you, let me also give you the connection of the So this is the blog that I want to share with you, 2013, and you will read this. And we had published it in 92. Milk as prevention of viral disease like TMV, HIV, and The Economist says so. Economist published an article on that. Remember the famous journal, famous economic journal. But that's not the important point. The point was that why do we wait for the Westerners to say something is useful without us rising to the occasion? So that's my question, that why can't we, where is the chat? Yes. Okay, let me put it here. So you can begin with this. This is a 2013 short note on my blog where I had put some references together, and you will look at it. And I would like that you will challenge what is said in this blog by your own arguments. You will provide new ways in which we can analyze this problem. This is just an illustration. These 3 weeks should help each one of you to ask fundamental questions, sometimes applied questions, and pursue them like a scholar. And if possible, and I would be very happy if you did that, if possible, publish short communications, 1 page, 1.5 page communication in good journals after doing your review, rigorous review. Doesn't matter if this is rejected, doesn't matter. Even the experience of getting rejection is very useful as a scholar, because there is no scholar in the world, including myself, whose papers have not been rejected first time. So it is not a shame that your paper will be rejected, but it might be accepted. Who knows? You try. So we would like you to aim very high in writing reviews, publishing them, acknowledging everybody's contribution, even oral communications as personal communication, being very ethical in acknowledging the sources. Don't ever quote, don't ever use anybody else's text without proper citation and without proper quotation mark. 3 or more than 3 consecutive words taken from any source without quotation mark and reference is plagiarism. Please remember that. It's a plagiarism. And 0 tolerance for plagiarism. Never do that. This cut and paste culture must end today for all of you and for all your friends and your network. This has done a great harm to our original thinking, because we tend to borrow ideas without acknowledging them properly. Obviously, you can't incorporate somebody else's language as a part of your narrative. You have to be very careful. 3 or more than 3 consecutive words taken from any source without quotation mark and citation. Just citation is not enough, because these are not your words, somebody else's word. So you have to put quotation mark around them. And a page number if it's a book, journal, whatever. And date of download if it's an online source, along with their reference number, site address. So please remember that we will follow the best ethical practices in dealing with the sources of knowledge, both oral and written. Oral means each other. If any one of us gives ideas to each other, we will acknowledge them. And we will acknowledge them both approvingly if they are good ideas, and if we don't agree, we will also acknowledge them if they raised a question in our mind, if they're worthy of acknowledgement. Sometimes there's an idea which triggers a thought in our mind, and we should then be very happy to source that because of this comment by so and so, I got interested in the subject, I did this, I further read about it, and then I discovered that this is how we can tackle this problem. So that trigger was very important. The trigger of curiosity will lead to good science, good experiments, good technologies. Department of Biotechnology and BIRAC, Biotechnology Industry Research Assistance Council, has worked together with SRISTI to create this program. We are very grateful to them. And this is the 7th program, as I said. Earlier, all of you would have come here, as it used to happen. But we are still trying that when the COVID-19 effects subside, if not together, some of you who have done good work would come in batches to our lab and experimentally validate your attempt to find science of farmers' practice. So recently there was a lot of migration, as you know, a lot of migrants who had come to the city had walked back, which was very painful, very, very painful. millions of people walking back home under great stress. So there were some students of Micah. I was talking to them yesterday, and they wanted to do some projects. So I asked them, why don't you map the mind of the migrants? Now, who are migrants? All of us are migrant actually. I was born in UP, in small village Gangagad in district Bulandshahr, and I'm now in Ahmedabad. So naturally, I've migrated from my village to Delhi, to Hisar, where I studied, and to Kanpur, where my father worked, and then here. So all of us are migrant. Some are voluntary migrant, opportunistic migrant, some are involuntary migrant. So this assignment given to them was that talk to the servants, talk to the maid, talk to the worker, talk to the driver. I think, Mega, you had given this assignment to them also?
Ji sir, they have submitted and I
Okay,
have
so let us review and pick up some unusual practices. Can you tonight send me some of those unusual practices selected by them?
no problem, sir.
So then I will like to discuss with them tomorrow or day after. So one of the lady who was working for 5 years in this family, Pinky, was interviewed by a student, Juhi of Micah. And you know what she mentioned? Very interesting. Something that I didn't realize, I must be honest with you. She said, when you drink water, you should sit down and drink water. When you take milk, you should stand up and take milk. Now, this was very clear because I didn't realize that. Though I know that in UP, people stand up near the shop and drink milk, but I thought that was an aberration. Generally, people should sit. But no, it seemed that there was a dynamics in the physiological process that a milk movement might be facilitated and be digested better. Now, this is an observation of an illiterate woman, and she made many other observations. There was a gardener who told another student that if you have fungal disease in the crop, mind you, root rot. Some of you might have been, how many of you have been to a farm, agricultural farm? Just some idea? Let me see if I can see you in the gallery view. And we are in the gallery view. Show non-video participants. All right. So now I can see. So just raise your hand and then I will find out. There is a raise hand option. Just raise hand, then I can see how many have been to the farm. There is a hand raise button, Mega, in this program.
They are doing it, sir.
Huh? Nahi hai?
Some of you, there are many who did that, sir.
Acha, acha. All right. So what happens? Root rot is a fungal disease. Many crops get affected. So what this farmer told us, I think Jagrup or what his name was, and he said, if you put asphatida in a poultice irrigation channel or alum fitkari in a poultice at the mouth of the irrigation channel where the water flows into, it will help. It will get mixed with the irrigation water and will be spread in the field and will help in controlling fungal disease. Now, this is an observation of an illiterate person. We use fungicides, toxic chemicals, but if the answer is so simple, and if it's low cost and it's extremely affordable, then it will be worthwhile finding the science of it, isn't it? So we have only a few minutes left before Dr. Shekhar Mande joins, so I will finish quickly. What I'm trying to say, my dear friends, is that this is an opportunity for you to intensify your curiosity about people's knowledge, to use the best possible science and build a bridge between formal and informal science. Ask yourself, how can we prove it wrong? The first response of any one of you will be, this is not possible, this is not correct. Okay, because science begins with the null hypothesis. Null hypothesis is opposite of what you want to prove, isn't it? So let's say the null hypothesis for this experiment will be asphatida does not help in controlling root rot, or alum solution does not help in controlling root rot. And then we'll try to prove it wrong using good statistics, good experimental design, proper observations with proper control Do you know the meaning of control? How many of you, anybody can explain what is the meaning of control? Riya, Saksham, Shweta, anybody? What is the meaning of control? Anybody, what is the meaning of control?
So to stop the growth.
No. Control
So-
in an experiment, when we say we should have treatment and control, what is the meaning of control?
So inhibit
This is for
something.
comparing.
Standard.
If you handle anything in your own,
हाँ बोलो हाँ श्वेता बोलो.
if you handle anything in your own way,
नहीं. हाँ मतलब control is where you are not applying the treatment but other conditions are similar. So let's say we have an experiment on a crop, then the date of the sowing, the variety, the agronomic practices, the manure, irrigation schedule, all of these will be same in the control as well as treatment. But we will only distinguish the 2 plots or 3 plots, depending upon how many treatments we have, in terms of with and without. So simple experiment is with and without. That is the first level of inquiry. We just do a quick experiment to see, does it make a difference? Then we set up a statistical design and replicated design. There are various models, random block design, there are different kinds of design that we can do. And then have more replications and then do the experiment and then derive the data and then try to prove through the standard error being less. So the treatment effect and the environment effect, we do ANOVA, analysis of variance. How many of you know analysis of variance? Anybody knows analysis of variance? ANOVA and MANOVA. So Professor Shekhar has already joined us. Wonderful, Professor. Welcome.
I just sent a small message to you.
Andy.
There's some event that unfortunately I will have to leave at 10:30 or something.
Yeah, so you will start now. Can we start now?
Yeah, and I don't have to make very detailed remarks, very small things.
No, because I want to tell them that you are among one of the few leaders in the country who takes a lot of pride in encouraging and highlighting the goodness of others. So there are leaders who will talk about what they have achieved, what they have contributed. But Professor Shekhar Mande, Director General CSIR, I welcome him very much to this conference, this program. He has kindly agreed to inaugurate this BIIS program where we have students, bright students from all over the country, from all parts of the country practically. And he takes great pride in highlighting the goodness of others, highlighting the contribution of other scientists. That's one great quality that I find in him. Second great quality that I find in him is that when he finds that something exciting is happening, he doesn't wait to be told, he goes to the field. So recently, I saw the pictures where he went to villages, interior villages in Himachal Pradesh, where first time Kesar was grown outside of Kashmir, and he, asphatida, we were talking about asphatida, how asphatida poultice put in the irrigation channel helps in controlling root rot, fungal disease. And Professor Shekhar Mande, we were just discussing that how young minds should do justice by being skeptic, but at the same time curious about the traditional knowledge of our society and subject it to scientific scrutiny. And if they find valid, then they should be able to say so boldly that this thing works very well, this is low cost, it is a viable solution. So each one of them would actually be taking up some grassroots knowledge, grassroots innovations, grassroots practices, and developing scientific protocol to test them, so that when they grow up, they will be our reignited minds of our society who will treat knowledge of people with greater respect, I would say, and also greater curiosity, so that the years of effort that our society has made in solving problem doesn't go waste and does not get neglected by the formal scientific system. So over to you, Professor Shekhar Mande. It's our privilege to have you with us, and I'm sure you will inspire and encourage the class in a manner that nobody else can. Welcome.
Yeah, Namaskar. Thank you so very much for bringing me to this platform, Professor Anil Gupta. I saw the list of the students who are attending this, that Excel file that you have sent me, and it's very impressive that people from all corners of the country have joined this particular program. And I can assure you that all of you will not feel disappointed. Professor Anil Gupta is by far the name in the country for having catapulted the grassroots innovation movement. And many of you are aware of his very famous Shodh Yatras that he does. And he has brought so much beautiful knowledge in front of all of us that many of us were actually not aware that people in their own small settings, in villages, in remote corners, how they lead their life very happily, very peacefully, and with the day-to-day innovations that we find it very difficult to even imagine sitting in our air-conditioned offices. But difficult to imagine that people can be so innovative in actual own surroundings. And that's thanks to Professor Gupta that all of us know much more about it than ever before. There are many different examples that how humanity has actually always coped up with challenges of the surroundings, right? I mean, we as human beings, starting say about 15,000 years ago, when we started forming our own small societies. Before that, of course, we were hunter-gatherers. Even then, we used to innovate. But as societies, the challenge is far more than living as the hunter-gatherers. I mean, I will not say more or less, a different kind of a challenge to live in a society than living at a hunter-gatherer. And for that, there's an acute need of innovations all around for societies to be able to live together in harmony and peace with not only with other societies, but also with nature. And there are many different examples, even within our own country, how we have done that. And the historical records of the excavated places, such as Mohenjo-daro, Harappa, or if you go in Gujarat, places like Lothal or Dholavira, are some fantastic examples of how 5,000 years ago people could innovate and make a society very bubbling society to live, where people probably were doing very active trade, people were pursuing arts, people were pursuing many different ways of modern human endeavor, even then at that time. So these fantastic examples of how innovations actually led to societies which were so prosperous even 5,000 years ago. And it has continued till today. There's been a very continuous tradition about all of that even today. Now, I'm told that this particular meeting, you will be mostly focusing on biotechnological aspects of innovations. Is that correct, Professor Doshi? Professor Gupta?
Yeah, biotechnological, life science aspect, broadly life science.
Yeah. So, and in the country also, there's another very great thing that happened that some years ago, Dr. Bhan, who was a very great leader who could see actually far in future what could be, he along with Dr. Renu Swarup came up with an idea that they should start this BIRAC. And BIRAC has literally catalyzed the biotech innovation system in the country. They have done some absolutely fascinating job. And when two organizations together, the one which is connected to grassroots, such as SRISTI, and when BIRAC, which is enabling or catalyzing these innovations, they come together, obviously we can expect miracles for the time to come. And I'm very glad that all of you have joined here to participate in those miraculous moments for the future. As I said in the beginning, you will all feel very enriched having attended this program at the end of it. The future of our people, the future of humanity is in your hands, and we do look forward to you innovating. Just on the side track, I want to mention a small thing. Professor Gupta mentioned about my visit to Himachal Pradesh. That was about 10 days ago, where I went to see the plantation of Asafoetida and Kesar in Himachal Pradesh. But also came back with a very refreshing trip from Lucknow for the last 2 days. And one of the objectives of going to Lucknow for me was to visit Ayodhya jail, not for any wrong reasons, but for the right reasons. That people who go to jail, they have an social stigma associated with them. You know, I mean, they're not accepted in their family once you go to jail. But as you also know, a lot of social stigma one can get over by simple economic means. If a person has a livelihood and starts earning one's own money, even a person in jail can actually try to remove that social stigma around that person himself or herself. And towards this, we actually went there to teach the jail inmates how do you make value-added products from waste flowers. You know, and then what we have done is we have taken the help of an NGO in Ayodhya. They collect waste flowers from all the temples in and around Ayodhya. They bring it to jail. And our scientists have gone there and taught them how do you make value-added products out of this. For example, one can make showpiece items out of flowers. For example, one can make agarbattis out of flowers because they have very good fragrance, many of these flowers. And for example, one can also make gulal out of this, natural gulal. And this is exactly what our scientists were teaching the jail inmates yesterday, in the hope that this will allow the inmates to have their own self-sustenance, even within the jail and when they come out. And to tell you very frankly, Professor Gupta, that one cannot feel elated or very satisfied by seeing the fruits of what we do in our laboratories reaching the direct beneficiaries in the field. I think that's the best of the real eureka moment for a scientist would be that to see that people on the ground are benefiting by what you do in your labs. That's a very, very emotional moment. And that actually is what many of us actually live for. I do hope that kind of enthusiasm is rubbed on all of you by Professor Gupta. He's a real champion of this kind of a thing, of how to connect with the real people on ground. And I'm pretty sure that all of you will feel enriched by his experience. Thank you very much for calling me here, Professor Gupta. Thank you so much for allowing me to be among the students today. And all the best to all the participants. Namaskar.
Thank you so much. Thank you so much. We realize the pressure on your time, so we will not ask you to stay longer. But certainly, if you have time, sometime to talk about science and the way young minds in your lab asked questions which you found very intriguing, some anecdotes of that kind will be very nice for our young mind. That how sometime even the senior scholars feel intrigued by the questions of the young mind and how that can trigger new discoveries and new research. So maybe whenever you have time, even in the evening, if you have time, 15, 20 minutes to talk about some such stories of how intriguing questions of young mind can lead to good experiments, good design, that will be very interesting. Thank you so much. Thank you so much. Thank you so much. We're grateful.